This is question 1 from the AS Paper 1 from 2017. 4 parts to the question, and the first 2 parts students do well at, but the second 2 parts students do dreadfully at. Let's read through it. It says figure 1 shows part of a DNA molecule. How many nucleotides are shown in figure 1? Well, most students got that correct. The answer, of course, was 8. Because each nucleotide is made up from deoxyribose, which is your pentose sugar there, a base and a phosphate. And you can see I've circled each of those. So in the 2 strands, there are 8 altogether. The next part of the question asks, name the type of bond labelled X in figure 1. Well, bond X is a phosphodiester bond. And of course, there's no way of guessing that. You would have just known that. It's simple recall. Part 3, however, asks, the enzymes DNA helicase and DNA polymerase are involved in DNA replication. Describe the function of each of these enzymes. So only 28% of students managed to get both marks here. DNA helicase breaks the hydrogen bonds between the bases. At A level, if you use words like DNA helicase unzips the DNA or unwinds the DNA, you will not get the marks. You need to be more specific than that, and you need to talk about the hydrogen bonds being broken. And again, between the bases. By saying between the nucleotides, do you mean the nucleotides parallel to or adjacent to? So that's why it's safer to say breaks the hydrogen bonds between bases. And the next one, DNA polymerase, well, that's what forms the phosphodiester bond between the nucleotides. Now, if you couldn't answer the previous question about bond X, you're not likely to remember that it's a phosphodiester bond. You could actually just get the mark by saying that DNA polymerase joins adjacent nucleotides in the sugar phosphate backbone. Using the terminology sugar phosphate backbone is really good because it enables you to describe really clearly where those bonds are. In your writing sometimes, when I read students' work, isn't always explicit which bonds are breaking and where they actually mean around that nucleotide molecule. But it was the next question where students performed particularly badly. It says adenosine triphosphate, ATP, is a nucleotide derivative. Well, what that actually means is that adenosine triphosphate was made from, originally, a nucleotide. And the next part of the question goes on to say, contrast the structures of ATP and a nucleotide found in DNA to give 2 differences. Well, when you get that contrast command word, remember, we've got to use the word and in all of our answers. We either use the word and, or you would be saying something like it's greater than or more than, which is a contrasting word in itself in the sentence. There were 3 possible answers you could have given. The first one was to just say that ATP has ribose and DNA nucleotide has deoxyribose. Next, you could have said that ATP has 3 phosphate groups. Remember, it's adenosine triphosphate, and DNA nucleotide has only 1 phosphate group in its sugar phosphate backbone. And the third possible mark you could have given was to say that ATP base is always adenine, and in DNA nucleotide, the base can also be cytosine, thymine, or guanine, or in other words, you could have just said it varies. So what are my top tips for this question in general? Well, as only 28% of students scored both marks for question 1, part 3, because they either used words like unwind or unzipped, or they just weren't explicitly clear about the sugar phosphate backbone or if the bonds existed between bases. Use proper A-level vocabulary, which of course unwind and unzip isn't. My next top tip goes back to the word contrast, which is our command word for that question. If you're answering a contrast word, then we need to be saying and comparing 2 sides of the statement. Also, don't confuse adenine, which is a base, with adenosine, which is adenine plus ribose. So adenine is one thing on its own, and adenosine has ribose added to it. Hence, that's where the first mark came from for saying that ATP has ribose and DNA nucleotide has deoxyribose. Remember, we know this stuff, and rereading the textbook isn't going to help. The only way to make progress is by having a look at these past paper questions, reading the examiner's reports, reading the mark scheme, and listening to these videos. Hope the video has been useful. Please do leave me a comment.